Hello there, here I am again. This makes it lecture number 16. Today I'm going to be talking about something called genetic expression and regulation. I'm hoping this won't be too long a lecture because I'm going to be quite frank with you. This subject gives me a headache. When we learn about genetics, when we learn about how we take the DNA in a cell and it goes through this process of transcription and translation, it makes proteins, those proteins produce traits. We have this concept called the central dogma of molecular genetics, and it sounds to me like if we have the gene, we have the trait. It's going step A, step B, step C, all the way to the end. Have the gene, have the trait. Well, it's just not that way. And so we have to talk about how much more complicated than that it is. And I want to make sure you understand how much more complicated it is without making it so complicated that you can't understand anything about it. And that's why it gives me a headache. Anyway, I'm going to be talking about how knowing we have the gene, knowing we have the capability of making the protein is not enough. Expression is whether or not a gene is actually being transcribed and translated and made into a protein, and regulation is whether or not we can turn that gene on or off. So just because the pea plant has got the right gene doesn't mean it's going to have purple flowers if there is some type of expression or regulation issue that has an impact on it. What do I have here on this page? Well, I have a couple of funnies. I'm not sure how funny they are, but we have one that looks like a family of cells, and we know that when a cell undergoes mitosis, it divides into two. We even call them daughter cells, and it makes a complete identical copy of the DNA. That does not mean that the cells that get made are going to be identical to the parent cells that made them, because they have all the same DNA, but that DNA is being regulated and expressed differently. So we have some parent cells here that are looking at Junior. It says he, which is surprising because it ought to be a daughter cell, right? Right? Okay. Anyway, Junior looks a little bit goth, maybe a little bit, just maybe just a little bit anti-establishment, I'm guessing. And then the parent cells are not too happy about that. He's differentiating from us, Rita. Anyway, that's dad's cell. And then over here we have apparently some DNA that in order to express itself, it has to sing. It says, yeah, I know it is weird, but this is how they expressed themselves. Okay. Anyway, just trying to be funny, I think, with those words. But like I said, this can get kind of complicated. Let's take a look. All right, well, here are a diagram and another funny. I put a funny in the second slide. That's not the way I usually do this. Anyway, a diagram and another funny to kind of re-emphasize the points that I was just making, and that is that even though all of the cells in the human body have the exact same complement of genes, and in fact there are 30,000 genes in the human genome, and they produce 90,000 traits. Even though they all have those same exact genes, these cells look very different. The cells in your heart do not look the same as the ones in your pancreas, or the ones in your lungs, or the ones in your brain. Oh my goodness, why are they so different from each other if they all contain the same genes? That is because those genes are regulated and expressed differently. And the funny here is it looks like what's going on is some cells that thought they were in the stomach are in the eyes, and so they're producing hydrochloric acid, which is something that's useful in the stomach, but not so much in the eyes. I don't know if that's funny or not, but it gives the impression that we might want to make sure that the right genes are turned on and off, that the right genes are expressed properly in different parts of the body. And this is showing that it's not just about where the cells are in the body, but when they are in the body in terms of what genes are getting expressed, what proteins are being made. Here we have a picture of the development of a fetus over 9 months, and clearly the genes that are being expressed at 2 months are not the same genes that are being expressed at 8 months, and we could take this on into childhood, adolescence, adulthood. The genes that are being expressed in my body right now certainly aren't the same ones that were being expressed when I was going through this process. So where the cells are makes a difference, and when they are in the development of the organism makes a huge difference. We really have to understand the complexity that can come about because of these regulation and expression issues. So if we want to give a good impression of how complicated something can be, why not make a big complicated list of all the different things that can vary within this process? This is a list of things that could be different when we have a gene and we're working toward making a protein from that gene. And everything has a question mark next to it because it could happen or it could not happen, or it could happen in an alternative way. So all of this idea that we have that once the gene is present and it's being made into a protein, we have the trait, that kind of has to go out the window. I'm really sorry about that. The details here I'm going to talk about as I go through this, but everything here, repressors, inducers, spliceosomes, protein processing, coenzymes, feedback, timing, these are all things that can vary in the process of that gene becoming expressed. So this is supposed to maybe give you a little bit of the headache that I'm having when I think about this kind of stuff. Okay, so just a little indication of the complexity here is I've got a picture that I used to use when I taught genetics at Columbia College, and I talked about genetic regulation. And when I first started teaching genetics here, there were 3 places that genetic regulation could take place. And this was in a genetics class. This wasn't in a human biology class. This was trying to learn about how these genes work. There's 3 places. The first one is during transcription, when we're making RNA from DNA. And we might be able to say, don't make it, or yes, do make it. So either the gene is being transcribed or it's not being transcribed. Boom, we've got some control over whether we're going to make the protein. The next thing that can happen was during something called RNA processing, which I talked about. This is inside the nucleus. After the messenger RNA transcript is made, it gets cut up and re-spliced back together. And depending on what the cell needs to make in terms of proteins, it might get spliced together in a slightly different way. So we might say there's alternative splicing, alternative processing. So we've got transcription, on or off, we've got alternative processing, and then finally we've got translation. And during translation, we're outside the nucleus, we've got the messenger RNA finds itself a ribosome, and then the transfer RNA is bringing over the amino acids and making them into a polypeptide chain. And there may be opportunities to turn that on or off, but there may also be opportunities to actually use those proteins in different ways, those enzymes in different ways. It may be possible to actually only translate a little bit of the transcript that's coming out. So these were the 3 places where we could have a little bit of control. And this was, like I said, when I first started teaching genetics. Now, here is a diagram from your current text. This is not a genetic text. This is a general biology text. This is from Concepts of Biology. And in this case, we're still talking about genetic expression, genetic regulation, but everything in the colored box here is an opportunity for that regulation. And so now we've gone from 3 spots that we need to think about it to 9 places where this regulation can take place. We're still looking at the cell, and we have some things that are going on inside the nucleus here, and we have some things that are going on outside the nucleus here. And we know transcription takes place inside the nucleus, and translation takes place outside the nucleus. So they're changing the colors depending on what type of dynamic is going on. Actually, I think what's happening is the blue involves DNA. The reddish orangish involves RNA, and then the purplish involves proteins. So we've still got 3 kind of general categories. They're not the same 3 things that I was talking about before, but they're broken up into all these different parts. This diagram right here is essentially everything I need to talk to you about in this lecture. And I really just want to, after talking about this for a while, I want you to understand all the complexity that can happen here in terms of whether or not a gene is actually going to be made into a protein or not. There's many, many steps along the way that we can speed things up, slow things down, stop things outright, and that's what I'm going to be talking about here. So first of all, we've got, it says here that there's something going on with the DNA in the chromatin. Remember, chromatin is when the DNA is wrapped around some proteins, it's packaged up into a chromosome. Well, it turns out that we can not express a gene by just not unpacking it, and that does happen sometimes. And that happens through chromatin remodeling, histone acetylation, and sometimes something called DNA methylation, where we cover the DNA with some little chemicals that keep it from being transcribed. This is basically opportunities to completely stop the DNA from being transcribed into RNA. So that's what chromatin modification is. The next group here, all those reddish orangish ones, those are things that happen during transcription. We can turn transcription on or off depending on whether we have the right activator or inducer. These are enzymes that might be involved in that. We can splice up the messenger RNA differently during RNA processing. I just talked about that. We can actually splice it just right and then not let it out of the nucleus, because if it doesn't get out of the nucleus, it's not going to find a ribosome. It will never be made into a protein. So there are ways to kind of bundle it up so it never gets out through those nuclear pores. Sometimes we'll let it out of the nuclear pores and then it'll just fall apart because it's not needed yet, so it doesn't get expressed yet. None of the ribosomes will accept it, and so then it just falls apart. That's called degradation of messenger RNA. So there we have the DNA, we have the RNA, and then the last bit is about translation and making proteins. And we can turn that on or off, or we can just leave it on, and then after we've made our protein, we can assemble it differently. We can put it together. It's kind of like the messenger RNA processing is splicing it together differently. We can actually make some polypeptides and then fold them or arrange them differently to make a completely different type of enzyme. So again, a way of regulating the expression of these genes. There's two more things here. There's the fact that the protein can degrade and fall apart, just like the RNA did. And then there is the fact that it's going to get transported to different places. Yeah, we can make it. We can make the protein, package it up, but depending on where it gets sent off to, it's going to have a completely different impact. So again, these are all places where it's not just about having the gene and making the protein. Boom, let's do it. Let's have that trait. It doesn't work that way. It works in different cells at different times all throughout the body. And that's good because we're complicated machines. Okay, so the next few slides are really just me repeating, reiterating, and maybe giving a little bit more detail about each of those 9 things that were in that last slide. So clearly that's an important slide if I'm going over them a little bit again. We're starting with the DNA. Remember that was, I think it was the blue color. And there were 3 things associated with that. Chromatin remodeling, I think, was what was mentioned in the box, but I also want to mention a couple of ways that chromatin gets remodeled. And there's a picture here that maybe it doesn't give us details that will help us understand it, but maybe it gives us some idea of how the chromatin, which is proteins plus DNA, might have an influence on whether it gets unpackaged or not. So what we have here is a diagram of blue DNA wrapped around these weird little purple spiky balls, which are the proteins that it's wrapped around. And together they make chromatin. And what we're looking at, I think, with this little zoom in here, is that, like I said, the blue DNA around the outside, and then the histones, which are what these little protein balls are made out of, have little pellets in the middle and then little tails on them. And sometimes if the little pellet is a little bigger or a little smaller, or if the little tail has chemicals on it, it will prevent that DNA from being unpackaged, or it will cause a part of the chromosome to become more likely to be unpackaged and transcribed. So the example we have here is what's called a histone acetylation, and what we have is we have these little green dots on the spiky protein balls, and those little green dots, these are acetyl groups, they're just carbons and hydrogens. They actually make it possible for the DNA to be unpackaged. That's what you're looking at there. Over on the left, it says unacetylated histones, and the DNA is all wrapped up and it cannot be transcribed. But on the right, the acetylated histones, they are kind of relaxing and letting the DNA out in between them, and as a result, we're going to be able to transcribe some of those genes. That's important. Then another thing here is called DNA methylation. There are actually groups, there are chemicals that can just bond to the DNA, which will prevent it from being seen by the DNA polymerase. So these methyl groups, really just, like I said, carbon and hydrogen, almost like the acetyl groups. If they get all over certain parts in the DNA, then the DNA polymerase won't be able to see it, won't be able to read it, won't be able to copy it, but then also won't be able to transcribe it into RNA. So anyway, just giving you the idea that there are things that happen long before we get to the point of actually making an RNA transcript that can prevent these genes from being expressed. Okay, so the next step down the line after we're done with unpackaging the DNA, we get it ready, we separate the strands, and you know that during transcription, RNA polymerase is going to jump onto one of those strands. This is a little bit of a zoom in, more details about what's going on there. And when I say more details, I'm like, wow, this is really complicated. So here's our DNA, our single strand of DNA, because it's been separated from the other strand. We're going to be reading it in this direction, and that reading of it has to start with a region called a promoter region. If that promoter region is in the right position, ain't gonna happen. Upstream from that, not downstream, but upstream from that, there are some little sites called enhancer sites, and if those enhancer sites aren't in the right position, not gonna happen. Each one of those enhancer sites has to have a little coenzyme attached to it called an activator. If those activators aren't in position, it ain't gonna happen. On top of that, we have to have this special DNA bending protein that's just the right shape so we can bend our enhancers with the activators on them all the way around to the promoter region. If that DNA bending protein isn't there, it's not gonna happen. Now we bend everything around and suddenly we have a group of fruit here. No, it's not fruit. It's a group of mediator proteins, and they're all just the right shape. So they tie the enhancers and the promoter region together. If any one of those little tiny purple grapes is missing, then the group of mediator proteins won't be just the right shape, and it's not gonna happen. Then finally, we have some general transcription factors, and I'm a broken record now. All of this stuff needs to be in just the right arrangement, all at the same time. There are so many different ways that we can turn this process off, that we can say, nope, we are not going to make this protein. We're not going to make this RNA transcript out of this gene. Let's move on to another gene. This whole mess of Bob has to look like this, just right, in order for this RNA synthesis to proceed. Wow, that's really complicated. So it says here that what I've been talking about is that transcription initiation might be curtailed, it might be stopped, or we're not ready for it. And on top of that, this diagram doesn't even try to explain this, but there might be more than one promoter region. There might be more than one promoter region, so that we're not going to express the whole gene because we're going to start on a promoter region that's a little bit downstream. We're only going to express part of that gene. Oh my goodness. This is just so complicated. I told you it gave me a headache. So this is where we're playing around with the transcription. Clearly complicated process. Okay, so now we got past the chromatin. Okay, we unpackaged the DNA. We got past all those acetyl groups, all those methyl groups. We unpackaged it. We separated the strands. We had all the parts that were required for doing transcription. We got our RNA polymerase into position, and we made our messenger RNA transcript. We made it. We got it. The next step in that process is RNA processing. The next step in that process is processing. That sounds silly, but that's what it is. And we know that that's splicing up the RNA and then, or cutting it up and then splicing the parts back together. Well, it turns out that that's also very complicated. We need a bunch of proteins to work together just right that are called spliceosomes, plus some other proteins. And they grab pieces of this RNA and they bring them together. And when they cut a piece out, they cut this piece out, you see, or maybe I should say this piece, because there it is. That's the excised intron. And they splice the ends together. But you can tell, again, once again, it has to be just the right size. Everything has to be just right in order for this process to take place. On top of that, if we're going to be making lots of cuts and putting splices back together, it can be like editing a video or a movie. And so when we do the splicing back together, we might do some alternative splicing where we could make a processed transcript that looked like this, or it could look like this. In the second one, we have cut number 3 out, and we have put in its place number 4. In the first one, we have that number 3. So that's going to be a different piece of messenger RNA that heads out of the nucleus and goes to the ribosome. So it's going to make a different polypeptide. We're going to make a different protein as a result of alternative processing. And that's what this says up here. Alternate RNA processing. Still very complicated. Okay, so we're almost there. Just getting to the end of this list. We have unpackaged the DNA. We dealt with the chromatin remodeling or acetylation or whatever's going on there. We unpackaged it. We separated the strands. We got the RNA polymerase into position. We made a transcript. We processed that transcript by cutting it up and splicing them together. And we got that transcript, that RNA, out of the nucleus. So now it needs to find a ribosome. It needs to get translated into a polypeptide. And then finally, it needs to be assembled into a final protein that is going to produce a trait either in this cell or in some cell nearby, or maybe one that needs to be transported off to through the circulatory system. But protein to trait is the last step. These are the things here on this little list that happen outside of the nucleus. We can simply degrade that messenger RNA before it finds a ribosome. If it doesn't need to be expressed, then it can be tagged and can fall apart before it even gets translated. We can interfere somehow with the ribosome. We can not have the right amino acids. We can not have the right transfer RNA. There are all kinds of things that can happen to slow that down, and that's called translation attenuation. We can have an issue with, after the polypeptides get made, they don't get assembled, they don't get folded properly, they don't get assembled together properly. We have a diagram on the right here that gives us a little more insight into that. Remember the RNA processing that took place, and we had an exon and the introns were cut out and the exons were kept. We're going to cut out the introns. Well, in this case, and this is often the case, these exons are parts of the final protein. So we need to make each of them into a polypeptide and then assemble them into this working, large, folded up structure. And in this case, exon 1, the green bit, is translated into domain 1, and then exon 2 is translated into domain 2, and then exon 3 is translated into domain 3. They all have to be there, and they all have to be assembled together properly in order to make this a functional protein. That's part of this protein processing. Even if all that goes perfect, and it doesn't have to, and we're not talking about necessarily making mistakes, but there's lots of opportunities to turn this process off. But even if it goes perfectly, we might end up making a protein that we don't need. Doop, it went through the whole process. Here it is. It's in the cell. Let's send it off to, we don't need it. Oh my goodness. Do you want to keep it around? Because it's an active biomolecule. It might actually have an impact on something. So we don't want to keep it around. So what happens is that it gets tagged with these little green, I don't know, they're not really green, but these little molecules called ubiquitins. It gets tagged, and then there's a big capsule structure called a proteasome that then surrounds it because it's tagged. It surrounds it and then breaks it apart. So this proteasome, this orangish capsule-looking thing, surrounds it and breaks it into the protein fragments, little peptides, and then it can be recycled, and that's great. So even if we make it all the way to the end, that doesn't mean protein is going to become trait. It means we can make it all the way to the end of making the protein and then go, guess we don't need it. And there's a way to deal with that too. There are ways to turn this process off, speed it up, slow it down, regulate it all the way along in all of these steps that we have to go through. So, is that the point of this whole lecture? Just to talk about how really very complicated this process is and how it's almost hopeless that we're going to get to the end and get the protein made just right and the trait expressed just right? Well, yes and no, because even though we can turn this whole process on or off, speed it up, slow it down at any point along the way, it still manages to follow some very, very predictable procedures. Very predictable. So predictable, in fact, that we can say that this fertilized frog egg is going to develop into this tiny swimming tadpole, which is then going to grow up into this large frog, which doesn't look anything like the other two, and in fact requires that all of these cells are expressing genes in all of a different bunch of different ways, so that the eyes develop properly and the skin develops properly and the lungs develop properly and everything follows the proper pattern. In fact, we call it pattern formation. Okay, when we have genes that come on and are activated in the process of development from a fertilized egg to an embryonic animal or even a plant to some extent, growing into an adult, following that pattern very, very specifically, we call that pattern formation or programmed gene expression. So even though it can be complicated, it follows predictable patterns. And it has to do with signals in the environment, with one cell developing first and then producing signals that are going to impact the second cell, which is going to produce signals that are going to impact the third cell. And so we can count the cells and know exactly how they're going to interact with each other. And that's what we call pattern formation. So here are a couple of examples of how this pattern formation might occur in an animal, in a developing animal. And humans are in fact animals. We would have a sperm, meets an unfertilized egg and becomes a zygote. The zygote has a nucleus, it has all the DNA in it, hasn't divided up, hasn't copied yet, but there's all kinds of stuff in the cytoplasm. In fact, a lot of that comes from the egg. You see that the sperm doesn't have much in it, just the nucleus. All the stuff in the cytoplasm comes from the egg, but by the time that fertilized egg divides into two cells, two separate cells, it's already divided up that stuff unevenly. These two cells are already different from each other, and they already have tasks to do, and they've already started this process of pattern formation. Because whatever these little chemicals are, the brownish circles and the greenish triangles, they are going to influence which genes get turned on and which genes get turned off, and how genes are regulated within this cell. So that's called cytoplasmic determination. The next step in this process is induction. And sometimes when we've got a cell that has just been created through mitosis, the cell that's right next to it, or maybe a cell that's nearby, is producing some chemicals that might have an influence on it. And so we have an early embryo here, says that it's 32 cells large. We got a bunch of cells down here near the bottom that are producing some chemicals that are sending signals to the other cells telling them what to do. And so we can zoom in on one of those cells, and these signal molecules are then binding to the membranes of the other cells and telling the DNA in the nucleus, yes, go ahead, transcribe this gene, or go ahead and send that transcript out to be translated. We're not looking at a lot of details here, but I want to give you a sense of how this can still result in very, very predictable regulated procedures. So, cytoplasmic determination and induction by nearby cells. Okay, last slide, and this one here is about fruit flies because I'm talking about pattern formation, and we have studied pattern formation in fruit flies like nothing else. We've done it because they are animals, and so we can learn about pattern formation in animals by studying fruit flies, even though they're not humans. But it would be unethical to study pattern formation in humans because it usually involves inducing mutations, causing mutations in cells and seeing what does that do to the adult organism. Well, that's okay with fruit flies, but not so much with people. That would be bad. So we use this kind of understanding in order to try to say this is how we believe things are happening in humans. So what do we have here? Well, as I said, with pattern formation, we have usually cells early on in embryonic development that have very predictable patterns in reactions with the cells around them. So we have something called an egg cell or an egg nucleus here, which has nurse cells around it and follicle cells around it. This is still unfertilized, but by the time that egg becomes fertilized, all of the chemicals in all of these little bits are arranged in a very specific manner so that from the fertilized egg, we get immediately a segmented embryo where we have a certain group of cells here, which can be distinguished from a certain group of cells here, which can be distinguished from a certain group of cells here. And if we track those, and we do this by cell mapping, if we track those up to the adult fruit fly, it's much bigger now. It's changed, it's added lots of cells. They turn out to be the parts of the embryo that become the head, the thorax, and the abdomen. The head is where all the sensory parts are. The thorax is where all the legs are and the wings, all the limbs, and the abdomen is where the gut is and the digestive bits and the reproductive parts and things of that nature. So it's following a very strong pattern of pattern formation here. You see that what we're looking at right here is 0.1 millimeters, and up here about that same scale is 0.5 millimeters. So it's going to increase 5 times, but all the patterns remain the same because all of these cells are interacting with each other. There's a kind of a freaky picture down in the left here because it's a good example of what happens when this pattern formation breaks down. There are genes that are in every single cell of the body, and they need to be expressed at just the right time in just the right place, or things can go terribly wrong. Well, in this case, we have taken a gene that is expressed in order to create a limb, a leg off of the thorax of the body, and it has become expressed in the head area around the formation of the antenna. On the left, we have a regular fruit fly head, and on the right, we have a mutant fruit fly head, where in fact, instead of growing antenna out of its face, it has grown legs because the wrong gene is being expressed in the wrong place. This seems like kind of a horror movie kind of thing. It's a fruit fly, so we can do these experiments without being too unethical with it. But it's a good example of how this genetic regulation has to be fine-tuned all the time. Are things going right? We need to make sure it's going right. Okay, we can continue. Are things going right? Are you going to make sure they're going right? We can continue. That's why all of these steps along the way are so important, because we can't have situations like this, or it's just going to result in a complete waste of biochemical effort and genes being expressed where they really shouldn't. So that's what we're looking at here. Anyway, So that was genetic expression and regulation. I'm done with this lecture, and remember, I'm gonna give you 3 more this week. So by the end of this week, you should be up to chapter 18, or I should say lecture number 18, and that's getting on toward almost being ready for our next exam. So I'll see you in class. Stay safe.